Olivier Ukunzwenimana, and a student from Ines Ruhengeri, Faculty of Health Science. Department is Biomedical, Laboratory Science, and the class is Year 3. Today, now on, I'm going to continue for the presentations of Applied Immunology, where I will be talking about I will be presenting the question from number 33 to number 36. The overview of what we are going to see is the principle of precipitation reaction in immunology. For the next question, we will look how the radioimmunology immunodiffusions RID test is performed. The next question will be the principle of ELISA techniques. We will end by differentiating the sandwich ELISA from the indirect ELISA. Then at the end, we'll put the reference where you can go and check if you need a more detailed information. By starting on 3 questions, the principle of precipitation reaction in immunology. The principle behind this precipitation reaction is all about the interaction between the specific antigen and their corresponding soluble antibody to form the precipitation or insoluble precipitate of antibody antigen complex. So here are the steps. There are 3 steps. In the first step, antigen and antibody, they interact together. The result will be the antigen-antibody complex. Remember, these are soluble antigen and which will interact with their specific antibody, also which are soluble. And the result will be the formation of antigen-antibody complex. In the second step, there will be the formation of lattice or network structure. This lattice structure is the structure formed by those complex antigen-antibody which formed, and they will form a lattice structure of a network structure when they reach to their optimal ratio or an equivalent zone. So in the last, those structure will become an insoluble precipitate. Here what we are saying, the insoluble precipitate formations. This diagram which is showing how the antigen interacts with the antibody to form an antigen-antibody complex. So for the next questions was all about how the RID test is performed. This test is done to measure the concentration of specific antigen or antibody in the sample for the purpose of diagnosis. The key steps is that 5.1. From the 1, the GIA preparation will be prepared. Here the agarose GIA is prepared and impregnated with a known concentration of the specific antibody or antigen that will be needed to be measured. Here we are together, and the sample diffusions will be in the small wells where they will be punching in the GIA, and the sample containing the target antigen or antibody will be added in the well. For the third step, there will be diffusion and precipitation. Here, the antigen or antibody in the sample will diffuse radially out from the wells into the GIA. As the counter, the corresponding antibody or antigen in the gel is formed an insoluble precipitin ring cycle. So for the measuring, those formed precipitin ring will be measured, its diameter will be measured and compared with the standard one. Here we will eradicate or sort out the target antigen antibody to know the concentration of it. For the next question was about the principle behind the ELISA techniques. As I've said before, when we are in immunology, we understand and we say ELISA, we understand the interaction between the specific antigen and their corresponding antibody. So this technique is done by applying 5 steps. The first step is where antigen is immobilized, antigen immobilization. Here, the target antigen is immobilized on the soluble surface, such as the micropyrites well, and this allows the antigen to be cultured and detected. The antibody bindings. The antibody comes and binds to those immobilized antibody on the second step. Then for third steps, enzymes coagulations. Here, antibody is linked with the enzyme such as HLP or AP, and this will generate the detectable signals from those mixture. Then substrate is added. A colorimetric or fluorescent substrate is added and which enzyme can be converted into measurable signal. This will be helping the signal to be measured. Then the signal detections. This will be done by fluorescent spectrometer or luminescent meter to detect the amount, to detect the intensities of signal generated from those enzyme substrate reactions. Yes, which and this will be the proportional to the amount of the target antigen present in that sample. Here is the diagram explaining more. And the last question was about how the standard sandwich ELISA differ from the indirect ELISA. We'll talk about the purpose, mechanism, procedures, applications, and sensitivity. First of all, when we talk about the purpose, the sandwich detects the specific antigen, whereas indirect detects the specific antibody. On the mechanism, the sandwich uses the culture antibody to bind to the antigens, whereas on the indirect ELISA, they cooperate with the antigen to bind to the antibody. For procedure, in sandwich ELISA, antigen is sandwiched between two antibodies, capture antibodies, and detect the antibodies. For indirect ELISA, primary antibody binds to the antigen and the secondary enzyme is linked with the antibody to detect it. They will require second enzyme link. That's why. And then for the application, sandwich ELISA will measure cytokines, hormones, or viral proteins for the purpose of diagnosing a given kind of disease. While for the indirect ELISA, they will be the diagnosis of infections by detecting the antibody. When it comes on the sensitivity, sandwich ELISA is high sensitive due to the dual recognition, while for the indirect is a relatively less sensitive than the sandwich ELISA. Hope you enjoyed. Thank you for counting with us. And here we put reference for you. You can check for these references when you need to know or to clarify other things, or you can comment down below in the comment so that we can look together for what next or for what is not being understood. Thank you. See you. Ciao, ciao.